Good evening, everybody. It's a big pleasure to have you here, to have you still here. I know it's a bit late, isn't it? But I will try my best to wake you up with the microscopic topic of today's micro talk we're having together here. So, speaking of microscopic things, microorganisms, or microbes, or germs, if you prefer the term. They're everywhere, aren't they? Have you noticed that? They're everywhere. They're in the soil, they are in the water, they are inside, outside of our bodies as well. And it's not a surprising fact, but humanity started to work to collaborate with them since the dawn of time. Like really before Christ, long time ago. And perhaps raise your hand if there is anybody here that would like to guess what were the first biotech products invented by humans. Yes? No? It's a no. Okay, I'm going for it then. Beer. Alcohol, in other words. All right. So yes, great answers and correct. I would say bread and wine, but still. At the end of the day, it's telling us we need more than just food in life as humans, don't we? And jokes aside, yes, we were able to work with them, with microbes, without even seeing them, because notoriously they're quite tiny. It wasn't, in fact, since the 17th century that we've started to visualize these tiny little bacterial cells, with the advent of microscope, more or less in the way we're intending it today. Cool, but that was just about it. So we were able to perhaps classify microorganisms based on shapes or motility, but really nothing more. Fast forward then to the 20th century, where we started to manipulate, to cultivate, and even more importantly, to select microbes at our own advantage in general. And what you can see here is a very old-fashioned technique, also known as petri dishes for microbiologists. In other words, we are having a container, can be plastic, can be glass. We fill that with medium, cultivation medium, so in other words, nutrients and space for microbes to grow, and we let them grow. So what you're seeing here, all these little dots are actual colonies originating from one single cell. And they live, they proliferate on top of this medium, on this solid medium, okay? So amongst all these dots, you can see some of those got actually a halo around them, a transparent halo around them, which indicates visually to us, the researcher, the microbiologist, that that specific colony is doing something very important for our experiments. For instance, I think that everybody in here at least once got tested for some sort of bacterial infection. So what's happening? You're going to your doctor, they got a swab of your saliva or blood, and then they send this sample, this unknown and let's say complex sample, to the lab for what? For screening. So for instance, in case of a bacterial infection, the answer you would like to get out of these methods is what kind of drugs should I take to get rid of this infection? So quite important answers we are in searching for. Unfortunately, such old-fashioned techniques are still used today. And I'm saying unfortunately because the reality is that these methods, we are going to call them conventional, traditional methods today, they come with disadvantages. Like one, they are solid. And this little detail actually is very important. When you want to screen a microbial population, fun fact, most of the microbes don't like this scenario, this cultivation media on solid cultivation medium. For different reasons. There are data out there actually showing us that if you're lucky by doing this, if you're lucky, you can get the 5% out of the total life of a sample. So, thumbs down. And fortunately, we moved on, okay? Because in this case, also, it's worth mentioning that microbes compete for life, really. They compete for space, they compete for nutrients, and as a result, you may overlook rare strains, slow-growing strains, because the fast-growing will be the first one to show up. Biased representation, bad. On top of that, they're kind of cost and time-consuming procedure. But overall, the question I got for you is like, how can we depict the complexity of life with such bias techniques, right? Because we clearly have some results, but can we do it better? Did we already? Answer is yes, we did. We're still executing complex experiments in the lab, but the laboratory became very, very small. So small that it can be compared with a coin, for instance. So it's an euro, it's not a Polish zloty, I apologize, but for the next time, you get the reference anyway. And you can also see inside of this lab on a chip device, which is very tiny device, as big as a inch, square inch. You can see some corridors, you can see some channels, you can see some rooms, and all these designs allowed us to execute experiments, multi-step, complex experiments. And on top of that, in the process of designing and fabricate such laboratories, we do have high degrees of customization. We can design our lab in the way we want to use it, in other words. So we're talking already about miniaturization. Keep this in mind for the next slides. And I told you we can execute experiments inside of these devices, and these experiments come in a droplet format. So let's talk about droplets. In this very, very linear and simple workflow, we can see 3 fundamental steps where we are using the droplets that we generate as individual experiments. So the droplet is sort of bioreactor, is a sort of private universe for the cell we encapsulate inside of the droplet. In fact, we do encapsulate one cell per droplet, since we are going for single-cell studies in this case. First off, we're making droplets, we put a cell per droplet, and then, with our droplets, we incubate our bacteria. So from one cell, you will have 2 cells, 4 cells, 8 cells, etc., etc., etc. Exponential growth. You will obtain a colony inside, confined in the droplet. And even more importantly, the third step, you see what's going on inside droplets, as we did with the microscope, and you select what's growing inside of droplets. So specifically, these yellow ones that are being selected from the pool of droplets are the equivalent of this one colony with transparent halo you've seen at the beginning. So we really, really, really want to find the needle in a haystack, this very tiny fraction of microbes that for some reasons are useful to us. And before we dive into these 3 fundamental steps, I think it's worth mentioning that at the first step of this protocol, we have already eliminated big disadvantages associated with conventional methods. Because, for instance, number 1, in this case, droplets are liquid, which is thumbs up for most microbes. They do prefer liquid medium. And also, via single-cell encapsulation, we prevent, basically, competition among strains. So there is quite higher chance, there are quite higher chances to detect every single strain, even the slow-growing ones, the strain that are very rare in the original sample. Overall, better results. And we said, number 1, let's make our experiment. What you're seeing here is first of a series of lab-on-a-chip device that you will see today. It's a droplet generator. Pretty straightforward. We're generating droplets. And we're doing it by mixing oil with the cultivation medium, which is, technically speaking, water with some nutrients for the microbes, allow for growth. And in a way that somehow, personally at least, it resembles a lot mayonnaise. I think we're all familiar with mayonnaise. I'm not a chef, but as long as I remember, you have to mix thoroughly a few components, like vegetable oil and lemon juice and some egg, okay? So you mix them thoroughly, and you get some very tiny aqueous droplets made out of lemon juice, of egg bits, in an oily background, let's say. And this is what's going on here. We're generating microscopic droplets at a very fast rate, so 2,000 droplets per second, namely, which, picture yourself, it's like 2,000 experiments per second being generated, unmatched numbers for conventional techniques. And the cherry on top is that all these droplets will have equal volume. And this equal volume is still there after the second step, so the incubation, where we let our bacteria grow. You can see here life of some sort inside the droplets, because again, the droplet is the universe for this microbe, so they can proliferate independently from all the others without competition. They're living happily there. And here you can see also empty droplets. And the secret here is that statistically, we prefer to have one single cell per droplet and screen some empty ones instead of having maybe the chance of 2 or 3 cells per droplet in the experiment. So we just dilute the sample a bit to achieve 99% single cell encapsulation. And I know that picoliters, micrometers are not like everyone's cup of tea, but perhaps you want to guess, just raise your hand again, it's not a test, how many droplets, and therefore how many experiments you could squeeze in a volume of a vodka shot. Dear Polish audience, I see some faces are waking up with the magic word. Right. 10,000. What's your name, sir? Nice to meet you, Tomek. Be with me. No, you're quite far off from the actual number. The actual number is more like 400 million droplets. So again, picture, in case of Petri dishes, the one we've seen at the beginning, they're like this big and this tall, you probably need this entire theater to put that into perspective. So again, miniaturization. And miniaturization also means cost savings most of the time, and this is especially the case. So okay, we have our droplets, we have life in it, we gotta move on. So we're going towards the culmination, towards this third step, the selection process of our experiments. Once again, here we have yet another chip device, and this chip is designed in a way that we're having optic fibers inside of this laboratory, capable of reading through the droplets. These that look like massive pillars are actually micro-optic fibers in the chip. They read the colors of the droplets in a very fast manner. And when the positive ones pass in the channel, we can detect that, but also trigger the sorting event in a way that we can pool the positive experiments. We can really retrieve that for whatever downstream applications you got in your mind. And let's picture this. For some reason, your droplets, you've got 1 million droplets to screen, and you're interested in the 0.1% positive droplets. So very tiny fraction, the needle in a haystack in a form of experiment. All your droplets are red, so every time a droplet is red, it's passing, you just say, no, I don't care, no, I don't care, no, I don't care. But when a pink one, which is your positive, pass in the channel, you will detect that pink color and you will say, I want that, I want to retrieve that. And we're doing this in a very fast manner. Once more, in this case, we're talking about 1,000 of droplets per second, 1,000 of experiments per second, pretty big numbers again. And the equivalent of you doing this, for instance, is like, let's picture this. Your grandma gave you a bag of candies, and they're like 1,000 candies. They're all red for some reason. You don't like these red ones, but you like the tiny fraction, the 10 pink candies in this bag. And the equivalent of you doing this is like, you got your bag, you open it, and in 1 second you have on one side the red candies you don't like, and on the other side you have your 10 pink candies ready to be eaten. So with that said, we had a look on a very, very powerful tool for single-cell studies. And to study what? Well, life, in other words, on a very microscopic level. We have very big advantages coming from such protocols. So we talked about miniaturization, cost savings. We're going very fast, which is usually good. But also, we can have a great chance to have less biased results, which is definitely the point number one of such approach. And such approach, these tiny droplets, were actually born within the academic environment not a long time ago, like 2 decades ago, more or less. And they already brought us quite big discoveries. in certain aspects within the academia, there were game changer, especially for single cell studies, genomic, transcriptomics, and so on. But they also found application within like the industrial sector, in pharmaceutical, cosmetic preparation. And what I do really hope personally, with all the premises we got here today, is I'd like to see such big discoveries being implemented even more in the real world scenario, for instance, in hospitals, where it can definitely and ultimately help ourselves, the people.